Good afternoon, everyone, and welcome. My name is Leslie Nelson Bernier, and I have the privilege of serving as the president of the UNC Health Foundation. And it's such a pleasure to have you here for our 11th in our Game Changers series to talk a little bit about COVID-19. As always, you're in for a huge treat today. And to kick us off and get the hour started, I'd like to turn things over to our CEO of UNC Health and Dean of the School of Medicine, Dr. Wesley Burks. And one thing, don't forget to put questions in the Q&A throughout the session today. We do hope this will be interactive in nature. So Dr. Burks, I'll turn it over to you to get us started.
So thank you, Leslie, and thank all of you for being on with us today. I'm excited to be joined in today's session by Dr. Nat Moorman. Nat's an associate professor of microbiology and immunology in the UNC School of Medicine, and he is the co-founder of our program called Rapidly Emerging Antiviral Drug Discovery Initiative, or READDI, and that's what we've asked Nat to come and talk about today. He'll go into much more depth on READDI and also be able to take your questions at large around COVID. Just to talk a little bit about, as we have before, some updates from the School of Medicine and UNC Health. First, our graduating class from the School of Medicine for 2021, that for them lived the last 15 months in a really different time in medical school, but did amazingly well. They had their virtual hooding ceremony with the other graduates from health sciences at Keenan Stadium recently on May the 14th. It was nice to gather together on that Friday afternoon. It was a really pretty blue sky day that we often have here in the spring. And it was nice for the students, particularly after, as I said, what they've been through for the last 15 months, to be able to get together. And it's one of those times in academic medicine that is a special celebration for all of us. And so we were thankful for the opportunity this year. Across UNC Health, from a COVID standpoint, we're continued to be in places that are encouraging. The numbers of patients that we have in the hospital, we've talked about those each time, are less than 75 now, which is radically different than we were when we would have talked, say, December, January, or early February, when we were up into the mid 500s. The numbers of people in our hospital are about what they were late last May. So it's been a year since they've been at this low level, which is really nice. As an example, here at the Med Center, we have less than 20 patients, which is pretty amazing for what we've seen over the last year. From an encouraging standpoint, there's a lot of work still going on related to vaccinations. UNC Health, again, at large, has given over 400,000 vaccinations since December 15th. We talked previously about my memories of having my first vaccination and how that took me back to my childhood and some of the early vaccine work that we got there. What has happened, though, in the last few weeks is that the adolescents, the ages 12 to 15, have been able to get their vaccinations. It's changed some at the Friday Center. The music's a little bit different. There was one young man that we got to see who wanted to get his immunization on his 12th birthday. So he was one of the first few in that time, so which is really nice to see. So just to be encouraging about those that have not been able to get your vaccination, hopefully that you can soon. So as we come back to Nat and talking, I just again want to thank you for being with us today. And like we have before, I want to start by asking him to talk a little bit about what personally this last year has been like for you, just how it's affected you personally and professionally. What's the year been like? So please go ahead.
Well, I'd imagine, like a lot of people here, it's been challenging. I have two teenage daughters. That's hard enough. Two teenage daughters at home in my workspace now, it makes it even more challenging. It's also been just been really hard to watch this pandemic sweep through the country and the world, and obviously the human lives lost and the economic consequences has been difficult. At the same time, I'm a virologist in the middle of a pandemic. It's in some ways something that I hope I'll never see again, but it has been really exciting to be here on UNC's campus throughout this. I'm sure you all have mentioned before, the UNC is the #1 school in the nation for coronavirus research, and to be a part of that team making the efforts to help develop the vaccines and therapeutics has also been a real privilege.
I can't imagine for your two adolescent daughters what the last year's been like trying to navigate school and friends and like that. So I know that's been a challenge and an opportunity. So as we get started to talk about the work you're doing, talk a little bit about how READDI came to be and then your involvement in it.
Sure, I actually put together a couple slides around that I'll share here. The backstory on READDI is interesting. Can you all see my slides? We actually didn't start READDI with the pandemic. We actually began doing the work that was behind READDI a little over 2.5 years ago. So I had spearheaded a program called the Infectious Disease Drug Discovery Program at UNC, or the ID3 at UNC. And that work was actually supported very early on by the Office of the Vice Chancellor for Research and by the Eshelman Institute for Innovation. So even before COVID-19 came to be, we were thinking about the problem of how can we make new antiviral drugs. So the goal of this research was actually to create broad spectrum antiviral drugs. So most antivirals now target a single virus, and that's rather inefficient. It'd be better if we could have a single drug that was active against many viruses. So our goal was to create broad spectrum antiviral drugs, and with a little bit of a twist on the way we did it. So rather than going after the virus itself, we focused on targeting enzymes in the infected cell as a way to prevent the replication of multiple viruses. Now, this is a pretty lofty goal. This is not easy work. And the other thing is it's very multidisciplinary. And I don't think you could do this kind of work at many other universities. But here at UNC, in fact, it was fairly straightforward to go around and collect the expertise that we needed to do this job. So when you're thinking about making broad spectrum antiviral drugs, we need a lot of different expertise domains. So we need virology, we need proteomics, bioinformatics, chemical biology, and drug discovery, of course. But right here at UNC on the School of Medicine campus, it's very easy to find all this expertise. So a group of virologists, a cast of characters here, including our own Ralph Barrick, Mark Heisey, and Stan Lemon, teamed up with Lee Graves and Jonathan Schisler over in pharmacology, who are experts in proteomics and bioinformatics. We brought in Tim Wilson and Bill Zurcher from the Structural Genomics Consortium here at UNC, as well as Ken Pearson Jiaodong Wang in the Center for Integrative Chemical Biology and Drug Discovery. So together, this group really did have all the expertise we needed just within a couple of blocks. They comprise 10 UNC faculty spanning 3 schools, 7 departments, and 3 centers, all brought together to focus on this one problem. And I'll just give you a brief overview of the research that was going on. So I mentioned before cellular enzymes or factors. The ones that we focused on were a class of cellular enzymes called kinases. But I wanted to show you this because I think beyond the expertise in the UNC faculty, it also highlights the resources we have here at UNC that makes this work possible. So we had 3 aims in this project. And the first was to identify those changes that different viruses caused in the cell. And here we took advantage of the Michael Hooker Proteomics Core that really has world-class expertise in quantifying these pathological changes in multiple disease states. But once we had identified those changes, we need to understand which were required for the replication of these different viruses. And again, just down the street in GMB, we had the Structural Genomics Consortium. And they had a collection of small molecule inhibitors of this particular class of cellular factors, kinases, that were very well characterized that we could use in our studies of virus replication. But of course, once we've identified that cellular factor that different viruses need to replicate, now we need to take that small molecule and turn it into a drug, a compound that we can use in humans. And for that, we relied on the expertise, as I had mentioned, at the Center for Integrative Chemical Biology and Drug Discovery, or I've had to practice this a lot, so I'm going to use it, the CICBDD. That's not an easy acronym, folks. That took a lot of work to get that to roll off the tongue. But the CICBDD actually has extensive experience developing small molecule therapeutics for multiple disease states and has been very successful in the oncology program. But again, bringing together this expertise and bringing together these resources that we have here on campus already really made it possible for us to start the work that ultimately would lead to READDI. But in the process of doing this work, I also just wanted to summarize, this program was successful as well. As I mentioned, this was the Creativity Hub Award founded by the Office of the Vice Chancellor and the Eshelman Innovation Institute. And what it's already giving us is a really unprecedented understanding of what viruses do to cells when they infect them and what they need to replicate. But it's also starting to identify new treatments for each of the viruses in our program, and including treatments for drug-resistant strains of viruses. And we also feel we're making great progress towards identifying therapeutics for viruses that lack treatments, including those with epidemic and pandemic potential. So this was sort of the work that was ongoing in December of 2019 prior to the emergence of COVID-19.
Nat, if you could talk a little bit more about how that work then developed, then taking that group of people that your collaborators, the cast of characters that you talk about, and then jumping from there, from that acronym into READDI. What was the process? How'd that happen?
Yeah, it's, you know, I've worked with Ralph Barrick, who I hope many of you are aware of, for quite some time. Ralph was a part of the ID3C. And then our other collaborator that we work with very closely and has worked with Ralph for a long time is Dr. Mark Heisey in the Department of Genetics. We worked together a lot. As we were sitting there, you know, in talking with Ralph, we had a good idea of the potential magnitude of COVID-19 prior to, I think, a general awareness of what was happening. We first heard about it from Ralph right at the end, just in very early 2020. And Ralph had said, hey, there's a new virus that's beginning to circulate, and this is something we need to keep our eyes on. And then as the following months progressed, it was clear from a virus standpoint that we were facing a potential pandemic of the magnitude we've experienced. And so Mark and Ralph and I were actually sitting in a conference room on one of the last days that we could be on campus, just kind of lamenting the fact that we knew what was going to happen, and we also knew that we weren't really prepared. And that was really the moment when we started to conceptualize the idea of READDI. So taking the same concept, broad spectrum antiviral drugs that we had been using to focus on existing viruses, and thinking about how we could turn that to face forward, to be proactive, to develop those broad spectrum antivirals in advance of the next pandemic.
So I wanted to ask a little bit more about, like, you're targeting what you described earlier, the enzymes that are involved in the replication of the virus, and then the drugs that you're going through the drug discovery process. How do they work on that enzyme? What happens that then changes the disease process?
Yeah, so that's a great question. So if you don't think about viruses every day, like I do, when a virus gets into a cell, it has to change that cell. So viruses aren't like bacteria. They can't replicate on their own. So a virus can only replicate when it's inside that cell. But as we know, viruses are really tiny, even compared to a cell, and they don't encode much material. They don't encode many of their own proteins. Instead, they rely on proteins that they steal from the cell. They hijack them, they change them, they make them do different things or turn them off and on when they shouldn't be off and on normally. So what we're targeting on, Wesley, is if we can see the things that the virus changes in the cell specifically, then we can develop small molecule inhibitors that basically just turn those changes back to the way they were in the uninfected cell. So the cell stays alive, but the virus doesn't have what it needs in order to replicate.
So how does that process and the drug discovery and then the drug development, how does that fit with the work that's going on from a vaccination standpoint for future diseases? Kind of put those two together, people have asked about.
Yeah, it's another really important point. As we've been developing READDI, some people have said, but well, Nat, we have vaccines now. Why do we need small molecules? You know, wouldn't one seem to preclude the other? And, you know, I hope that in the future we can develop vaccines just as quickly as we did for COVID-19. But the thing that's important to remember is from February of 2020 until recently, there were no options. You know, we have to, when a new virus emerges, we require the sequence of that virus and an understanding of that specific virus to develop a vaccine. And in the meantime, there's a window where we currently don't have any therapies, particularly for a new virus. And that's where small molecule drugs come into the picture, particularly small molecules that we know are active against entire families of viruses. So we don't have to know what the next virus is per se. As soon as we figure out what virus family it's from, what we want to be able to do is go to the shelf and pull out our broad spectrum drug that inhibits that entire family and immediately start using that drug to treat and hopefully prevent disease until the vaccines are available.
Okay. So it kind of related to that, but some have asked, and I'll try to summarize the question. If you take the family of the coronaviruses, the enzymes that you'll target will be kind of in the same family. And so if you target enzyme A, doesn't really matter what virus it is, they all have that same mechanism. Okay, so now take a rhinovirus, a virus that causes normal common URIs, particularly in children. Then the enzymes that are needed for that versus the enzymes that are needed for the coronavirus family, are they similar, like vastly different? Talk about that space.
Yeah, great question. And the answer is we don't know. And that's some of that foundational work that READDI's doing, and that READDI has to do. So these types of changes haven't been discovered on a global basis. So there's some foundational knowledge that's needed to drive this process. But what I can say is the work that we've already done shows that for a given family of viruses, those changes to the cell are very similar. And targeting those changes can inhibit the replication of multiple members in that family. So we do think that it's very feasible to make broadly active drugs against a family of viruses. But I think when we start to think about different virus families, you know, we'll probably need to have a different drug that will target that virus family. And at READDI, what we're focused on are these virus families that really have high potential to cause widespread viral outbreaks, epidemics, and pandemics like the coronaviruses.
Right. Okay. So there is one drug that's being used right now. There's another couple of drugs that are in development. Talk a little bit about how those drugs, like their mechanism, how they work, and how that compares to what your approach is here.
Yeah, so I think probably the best known antiviral drug for COVID-19 is remdesivir. I think it's interesting in the context of what I'll tell you in just a moment about READDI to remember the history of that drug as well. So the mechanism of action, to your first point, is it directly targets the virus itself. It targets the enzyme the virus uses to replicate itself. I think the other point that I like to bring up about remdesivir when we think about being proactive, and that people sometimes think, well, remdesivir was available right when or soon after COVID-19 began to spread. It wasn't widely available, but we had already identified it. And it's important to remember the backstory on remdesivir. It's actually a drug that's been in development for many, many years, going back quite a long time. And it's actually, you know, I'd hate to say it with Ralph in the room because I'd never hear the end of it. But if it weren't for Ralph Barrick, we wouldn't have been as prepared as we were. Ralph thought to go out and test remdesivir against the coronaviruses. And that work had already been done when COVID-19 emerged. So he could rapidly transition to COVID-19 so that it could be developed as quickly as it could. So in context of targets, most antiviral drug development work to date has focused on the virus itself, which really only encodes a limited number of proteins and thus targets.
Yeah.
While READDI's interested in those as well, its real focus is on cellular factors. So the 30,000 or so proteins in our cells that viruses might need to replicate.
Okay, good. There's been a lot in the news in the last 72 hours about the origins of the coronavirus in China and whether it came out of animals, the lab, and there's a whole lot more to come out of that, so that we'll have better information and know what the real story might be. But just some thoughts that you might have for the participants today. Your thoughts, I know you have a lot about what the origin of the outbreak has been.
I think the idea that this is a virus that has jumped from an animal reservoir into humans is clearly the most logical argument that's out there. This is how coronaviruses routinely emerge from animals into humans. I've seen absolutely no evidence at all to suggest that the virus was created in a lab. There is no data to back that up. How that virus initial transmission event occurred, we don't know right now. There are multiple theories out there, but again, to the main point, I think the most important point is that this is a virus that probably was in an animal reservoir and somehow or other came into contact with humans and then began to spread through the human population. Obviously the past year, we've heard a lot of conspiracy theories about these viruses. And I understand that may be easier for some people to accept, that's how something like this came to be. But in fact, it's bad luck. It's a virus that was in an animal that made its way to people.
So people have speculated, obviously, about that, but also about what the next pandemic might be. So we've talked about there are some viruses in West Africa that people had concerns about. They fortunately were more localized. But what are the 2-3 things that if you were laying awake at night would concern you most about, like that next pandemic and whenever, and hopefully it won't happen, but there are some things that y'all work with that are more likely than others to cause a pandemic?
Yeah, we spend quite a lot of time thinking about the next virus. It could easily be a virus that we've never heard of, a virus that doesn't yet exist, that's out in an animal reservoir we haven't seen, but we've certainly seen viruses that have the potential to make the shift into humans and have massive viral outbreaks. The virus that I hope people still remember is a virus from the Flavivirus family called Zika virus, which did just that a few years ago. And if you recall, swept through most of the Western Hemisphere. But there are other viruses out there. In addition to just in the Flavivirus family alone, we saw something similar. People will still recall West Nile virus, but there's viruses like Powassan virus that are still out there. But other virus families like the alpha viruses, I think people tend to not recall a virus called chikungunya virus that, just like Zika, swept through the Western Hemisphere a few years ago, but other viruses in that family that have, severe impact on human health. Venezuelan equine encephalitis virus, Eastern equine encephalitis virus is a virus we've been hearing a lot about lately. So those are some of the virus families, the coronaviruses, the flaviviruses, the alpha viruses, among others, that we spend a lot of time thinking about.
Okay. So enough to keep the rest of us awake at night when we're thinking. So, one of the panelists has some questions I want to come to in just a second, but one in particular on the drugs. The question is, are your molecular drugs able to be given orally, or they have to be given as an infusion like is done with remdesivir? Where's the direction that you're going?
Well, that is a fantastic question. So since our goal is to do the work ahead of time to have these drugs, we want to make drugs that are compatible with widespread use and access in the context of a pandemic. So all of the drugs, our number one goal is to have orally bioavailable drugs. And there's a couple of different reasons. One, it's much easier to transport drugs in a pill or a capsule or a tablet rapidly to wherever they're needed. Two, it's much easier to ship those drugs, not just here in the United States, but around the world. You know, pandemics are not local. So we need to be able to get the drugs that we seek to make out, not just to countries like the United States and the EU, but to developing nations as well, where there may not be the infrastructure to store drugs that would need refrigeration, or to administer drugs that would require IV injection. So orally bioavailable is our complete focus.
Okay, so a couple of questions that have been asked about. One, it was the School of Medicine requires students to be vaccinated, both for our students as well as our faculty and staff, along with the university. We're really strongly encouraging them to do so. We feel like that's the right approach right now. I anticipate in another couple of years that may be different, but for right now, the best approach for us is to really strongly encourage people to get the vaccination. Another question around the vaccination is people that were vaccinated for next winter, fall and winter, you know, when they get a flu shot, will it be a combination? Will we need to have a COVID booster? There are people working on the combinations. Now, what I do want to ask you about is what your thoughts are right now on the likelihood of all of us or certain groups of us needing a COVID immunization booster by fall or winter?
It's a good question. There's a lot of work going on right now, particularly in Ralph and Mark's labs, looking at how the current vaccines protect against the variant strains that you hear so much about. You know, there are several variant strains of concern. The good news is all the data that I've seen so far says that the existing vaccines are effective against the new variants that have been arising. We hope that that continues to be the case. But the other, you know, we can take some comfort in the fact that should there be a variant that breaks through the current vaccines, the new vaccine technologies, in particular the mRNA vaccines, provide a means to rapidly pivot and change the composition of those vaccines to provide protection. So my personal take on it is that right now, the data says the current vaccine will hold. We know that we may see some waning of protection over time, or potentially a new variant arise. And in either case, I think it's a booster, you know, be it annually or even more frequently or less frequently, may be in order, much like the flu shot, as you had mentioned before.
Yeah, it'll be interesting, obviously, for all of us in 10 and 20 years, the big takeaways out of the past 15 months from a scientific standpoint. mRNA vaccines have been around for a couple of decades, but really had never gotten the support until now to like take them through phase 3 and then as a drug. And what people predicted and how they work. And that's like one of the many, I think, successes that have happened in the midst of all the craziness in the last year. I'd probably, if we'd asked you or Ralph or Stan, you know, 2 years ago, could we have a vaccine, not tell you what kind, in like 9 months after it started, you know, I think all of us would have gone like, I don't know, that sounds pretty quick, but it got done, and which is to me amazing. So another question is around someone who that received both their immunizations, so they're a few weeks out from that, and they come in direct contact with an infected person, will they become ill? So just your thoughts around that.
Yeah, I think the thing to remember when you hear about vaccine efficacy is those numbers, those headline numbers that we hear, are about reducing severe disease and hospitalization in particular. So think back to a normal time when we actually went outside enough to worry about the flu and we had to get our flu shots. You know, the flu shot is a good guess at what's coming next, right? And remember that they may not always get it right, but even if it was a little bit wrong, the flu shot still gave you protection from really severe flu or hospitalization or death. You can kind of use that same mental model when you think about your COVID-19 vaccine. You may get sick. It could happen. The numbers say it's actually fairly rare that you would be sick enough to have, even take a day off. But what is very clear is that even if you were to become infected, your odds of developing severe disease that would require advanced medical treatment or hospitalization are very low.
Yeah, the statistics on that are pretty amazing. Like there's so many zeros in front of it that it's hard to fathom.
It really is. I mean, the nice thing about these, the way I think about it, the way I've explained it to friends and family is, even if you got sick in a normal world, you probably wouldn't notice.
Yeah. So what the effect of the mRNA vaccine and the process we just talked about and how rapidly they were developed and they actually work, hey, what's the impact of that on the yearly flu vaccine? You think the technology will switch to that for some, or it's not a good, is it not a good process to develop that yearly flu vaccine because of the guessing that you talked about earlier?
Yeah, Wesley, I'm not sure. I haven't seen much about the plans to use mRNA vaccine technology for influenza specifically. What I can comment on is this has really, in some ways, revived vaccinology. Now, as you mentioned, this has been an approach to making vaccines that's been considered for quite some time. And with the success of these vaccines, I've seen a lot of different vaccine strategies now that before would have been very challenging. Now people are thinking that the mRNA vaccine technology may be a way to develop vaccines in these difficult spaces.
Okay. So there's another question that came about someone on immunotherapy for autoimmune disease and their B cells being down, and how long should they wait after vaccination, or what's the effect of vaccination on their treatment?
So I am, don't think this is a cop-out. I am a PhD scientist. Right. So the doctor here means that I work in a lab. I would say that this is a very specific medical question. And for the answer to this, I would certainly refer you to your physician. That's a very important question that was asked, and it does have some very specific considerations, but I would be remiss to give you any specific medical advice.
Good answer. So thank you. So we're going to segue now. You've got some more slides to share, and then we want to do that, and then we'll take some more questions.
Excellent. Let me pull them up. So I'd like to just take the opportunity to tell you a little bit more about READDI itself. So we spoke a little bit about the precursor program, but as I had mentioned before, the drive to found READDI came from our experience working with the ID3 at UNC, where we had seen success, but to take that and to shift it forward into a proactive standpoint. And even it was a good idea, I think, when we made that decision, but over the past year, it's become even more important in my mind. Because as we start to have the luxury of reflecting on what's been happening over the past year, we really need to change the way we approach thinking about viral epidemics and pandemics. Just in the 21st century alone, a lot of people don't remember, we've already had at least 5 viruses emerge to cause widespread viral outbreaks. And as we continue to expand humans, where we live, changes in climate, we're coming into more and more contact with viruses like the strange words I said before, Mayaro virus, Powassan, and chikungunya. More and more people are encountering viruses like these. And that really increases the chances of the next pandemic occurring. I'll skip the depressing numbers about the cost of pandemics. I think at this point in time, we all have a very good idea of exactly how damaging they can be. And we're also fairly aware that there are some system failures. Not everything went the way we would have hoped it would have gone in terms of our reaction and our response to COVID-19. And so we think that because of the increasing frequency And because we found some issues with the way we plan now, we need to come up with a better solution. But I want to first just talk about a couple of the major issues that we've seen that have prevented pandemic preparedness. After that first slide, I hope you're thinking, well, it's obvious we need to do this work. Why wasn't this work done in 2018 and 2017? And it's really because our system for doing this kind of work isn't optimized for these kinds of viruses. Pharma companies typically don't invest in drugs, small molecules for pandemic virus families. It doesn't make pharma companies bad, it makes them a business. You can't spend the money, time, and resources needed to make a drug for a virus that doesn't exist yet. There's just no market force to drive that development. Now, the NIH does fund this kind of research, but it's really more at a project-based level, not at the broad level that's needed to drive forward drug development. And even where the government does invest, it's typically in what we call a panic neglect cycle. So lots of resources available when a bad event is occurring. And as that event fades into our memories, that funding tends to fade away with it. So we feel that we've really got to rethink this system, the ecosystem that funds and develops antiviral drugs. And that's really where we feel the solution is READDI, where we're seeking to bring together the best scientific and business minds to proactively develop the antiviral drugs we need before that next virus emerges, because we know it's going to happen. So we need to do the work now. So we've developed READDI as a public-private partnership. And I want to put that in the frame of other organizations that seek to fill a high unmet medical need where there's really challenging market forces that get in the way. You may have heard of the Coalition for Epidemic Preparedness, or CEPI. So this is another public-private partnership that's aimed at making vaccines for high-risk virus families. CARB-X is a similar organization seeking to make antibiotics for antibiotic-resistant bacteria. And READDI sits in that same space to fill that need for these antiviral drugs where market forces pose strong headwinds. READDI operates in three functional domains. The first is accelerating antiviral drug development. So again, finding the small molecules that will inhibit virus replication, and funding and facilitating projects across all stages of development. The second really seeks to address those market forces where READDI acts to aggregate and advance existing assets. So up until about 20 or 30 years ago, most major pharmaceutical companies, in fact, had antiviral programs, but they weren't very profitable. So most major pharmaceuticals have shuttered their antiviral programs. But in the process, they've left behind some really promising compounds. I like to think that they sit in a giant vault somewhere at a drug company. And what we're doing is we're asking drug companies to let us take those compounds out of the vault and keep developing them to make them into a useful drug for use in people. The second problem is that a lot of academics, while they still stay in this space, academic research only goes so far. You can find the target, you can find the new small molecule, but those later stages of drug development are really time and resource intensive. And so bringing together the business partners together with the academics to help drive those compounds forward. And the third area of activity is expediting treatment availability. So we can spend all the time we want making antiviral drugs, but if we can't have those compounds ready for use when that next pandemic arises, then we haven't actually accomplished our mission. So we're already working with partners to develop manufacturing plans, regulatory plans for approval, and preparing clinical trial plans for both safety and efficacy testing immediately when the next pandemic occurs. So across those three domains, READDI acts in two roles. One is as a funder, and that's largely on the initial stages through phase 1 human safety testing, but then also as a facilitator, helping drive those compounds forward through licensing deals, developing the clinical trial sites, putting our private sector partners in place to make sure that the drugs continue forward. This isn't theoretical, I would like to point out. So we've been very busy in the past 15 months or so. We're already working with multiple partners, not just academics like ourselves, but also partners in industry and with other strategic partners who can bring together not just the specific discovery skills and development skills, but also the business skills that we need to make READDI a sustainable and successful organization. We already have over 20 active projects going on with partners from around the world, not just here in the United States. And all of those projects are focused on virus families that have high epidemic or pandemic potential. And I'll just give the one example here of a project Ralph Barrick is leading. I like to show this picture because I haven't seen Ralph at the bench in a lab in a long, long time. So he gets a chuckle out of this one. But a project Ralph's leading where we're looking to repurpose inhibitors of cellular enzymes called kinases for use as COVID-19 antiviral drugs. So putting all this together that I've told you, it's really all driven by a vision we have for the next pandemic. We know it's a matter of if, or it's not a matter of if, it's just a matter of when. And so we want to have a different outcome so that we don't have to go through this version of a pandemic like we just have. Our goal is that on day one, we will already have effective antiviral therapies established and developed through phase 1 human safety testing. We'll already have had the phase 2 and phase 3 efficacy trials designed with plans in place for how they will be accomplished. We'll have our manufacturing plans established. And together, all of this will really help facilitate that global response and the equitable distribution of these drugs that will be needed to stop that pandemic in its tracks before it can spread around the world. And so with that, I will stop and kick it back to you, Wesley.
It's amazing what y'all been able to put together, not only within the broader university community, but really worldwide to work together. So I know it's an amazing opportunity for you, and I know a lot of good things are going to come out of it. If you reflect back on the last year, you think, what are like the biggest surprises that you maybe were unanticipated? And as I ask that, like for me personally, and through the healthcare part and the school part, how quickly people were able to adapt to kind of an emergency management mentality, particularly in healthcare, that we just had to have people working in places they hadn't worked before, to trust people to do things that they hadn't been able to do before, and quickly make decisions that we didn't have weeks and months to try to do that. And my hope is that most of that will continue on post, the pandemic and the coronaviruses. I don't know that I would have hoped each of those would have happened in healthcare, 18 months ago, but it's been a nice encouragement as well as surprise, particularly on the how well people have trusted each other. You're going to do your role and I'm doing my role, and we're going to work together. We both can't do all of it, but together, if you're doing yours and I'm doing mine, we can do this. And people had that kind of mentality, and it worked. And for many of the researchers, there was a really singular focus on, like, we know what we need to do, and y'all spent an inordinate number of hours doing it. But anyway, long-winded question. So what's the biggest surprise for you in the last year?
Well, let's see, the biggest surprise. I would echo your comments on the medical side, on the research side. I don't know that it was a surprise, but we've never seen that kind of multidisciplinary collaboration and effort have to come together in such a short time. And it really was pretty amazing to watch the entire School of Medicine for sure, but really across all the different schools here on campus, people bring together their different skill sets with the attitude of, how can we help? What can we do to make this happen? We want to be a part of the solution. So I think, I don't know that I would say surprised, but it was certainly a pretty fantastic thing to see people come together. I do think another surprise of the past year, call me an optimist, but I was surprised we weren't more prepared. As a virologist, we think a lot about viral epidemics and pandemics. I was surprised that this is the way this pandemic occurred, right? We've all read, as a virologist, the great influenza. We think a lot academically about epidemics and pandemics. So I was surprised that our response was not better. I was very grateful that our response here at UNC was fantastic and was really a team-based effort. And I think my biggest takeaway is really that founding principle behind READDI. We need to do the work now to make sure that we're prepared for the next pandemic when it arrives.
Okay, so a couple more questions have come in on the chat. One of them would be the question is it accurate to say that the purpose of antivirals is to mitigate effects of diseases like COVID-19 until vaccines are available?
Yes, that's certainly one purpose. It's where until that vaccine is available, you've got to have some kind of treatment in place. And so in that, I think the 9 to 10 months that we saw this time, hopefully that time can become shorter, but I think it's important to remember that that is a fairly miraculous turnaround time for a vaccine in any state. So in that time frame. And then the other place where antivirals play another role is not everyone in the population will be able to receive a vaccine due to underlying health conditions, medical issues. And so those people need treatments and protection as well. And small molecule antiviral drugs can fill that space.
And there'll be some viruses that you need treatment for that won't be so extensive that you have to have a vaccine.
Exactly.
In that way. So another question asked about phase 2 and phase 3 trials and how they're funded. And so just to for the broader group, phase 1 really is about safety first. Phase 2 is a little bit more about safety, and then it begins to look at efficacy. And then phase 3 is to fill in the blank spaces around safety, but really look at efficacy, so that the 30,000 numbers people talk about with the vaccine development, that's in that space of that phase 1, 2, 3. So you alluded to it a little bit, but talk about how phase 2 and 3 are typically developed.
Well, so when we think about phase 2 and phase 3, we actually get into some interesting space because we're trying to make plans for SARS-CoV-4 and SARS-CoV-5 or COVID 2026. So it's hard, we can't right now set up we couldn't run a clinical trial because that virus may not exist yet. But what we can do is, again, really leverage the expertise here at UNC, where we have clinical trial sites that are set up, not just in the United States, but around the world in places where new viruses routinely emerge. To have the plans in place, to have the sites designated where we could start these trials, and to start to think about what would that trial look like if it was a respiratory disease, if it was an arbovirus that's spread by a mosquito. How would we design those trials? What would our enrollment criteria need to be, and what would our broad-based efficacy outcomes look like? That's all work we can do in advance for families of viruses.
Ned, if you go back 18 months, you were headed in a certain direction for you and the people you work with from a research standpoint. How has COVID-19 altered that direction, or is it just accelerated where you were going?
That's a very interesting question. So I've always been interested in the way viruses interact with cells. And I've always had a focus throughout my career on how I could use the information that I gain in the lab to develop a new therapeutic treatment. I think it's really helped crystallize in my mind just how useful the information we develop in the lab can be. How we can take we can satisfy our innate curiosity to understand how things work and how viruses replicate, but bringing together those resources lets us turn those into new medicines or start us down that road. So I think I wouldn't say that it's necessarily changed the way that I've approached things, but it's helped me go out and find the resources and develop the resources that I need to really drive the basic science discovery that I love forward into new therapeutics.
So one of the questions and encouragement for you that is talking about that, just how much they've enjoyed what you've been presenting today, and the somewhat lack of recognition nationally for all the research being done here. It's been done for a few decades here, but now that in light of COVID, people know what's going on here. So you've been in the middle of that change. How's it feel?
I think I would first off say that we are very lucky here with one of the co-founders of READDI, again, Dr. Ralph Barrick. Ralph and his team have been at the forefront of coronavirus research for many, many, many years. Ralph probably wouldn't like the number of manys I just said there, but it's been 25 or 30 years. It has been fantastic to see Ralph and others on campus doing this important work get the recognition that they really deserve. We're very excited that we think in general, the UNC virology community, together with the medicinal chemistry community and the broader research community in general, getting the recognition that I've always felt it absolutely deserves. When it comes to this space, I can say with a straight face that I think that UNC is second to none. And to see people recognize that as well has been very rewarding.
Good. I just, even on the UNC health part, I've told so many people that for me personally, the comfort level that I had and to be able to talk to the physicians in global health and the work that they're doing here and nationally, and to listen to what y'all are doing and the expertise that you have, to know that like we know the latest stuff and we can make good decisions knowing the latest information. Like that's way different. I mean, it's a comfort level that is hard to overestimate how I felt in the middle of that with UNC Health. So I'm going to go back a couple of 3 decades. So at some point you decided you wanted to study viruses. So like based on what you've already told the group today, how'd you get there? Why viruses opposed to whatever?
Yeah, I am one of those strange people who just became absolutely fascinated with stuff like this at a very young age. I think it sounds cliche, but I wrote a report in junior high on genetic engineering and DNA and fell in love with that. I mean, I think, Wesley, you'll probably get this from running a lab as well. Prior to that, I wanted to be a mechanic. And actually the two are not a whole lot different in some regards, but yeah. I fell in love with genetic engineering, the concept of molecular work very early on. In high school, had some fantastic teachers who, taught me about viruses, and that completely captured my attention. I am probably the biggest nerd I know. I applied to my undergrad in the microbiology program specifically to become a virologist. Learned molecular skills during my undergrad and went on from there and never thought about anything else.
So it's one that's an amazing story for you personally, but I think it's always fascinating to find out how the people that we work with here, how they got to where they are. And some early on knew like, this is where I'm going, and others, it was kind of a windy road and they made a few detours along the way, but have wound up at some really good places. So I'll turn back to Leslie now and additional questions, thoughts that she has as we close the hour.
Sure. Thank you so much, Dr. Burks. I really appreciate it. And thank you, Nat. This has just been such a fascinating presentation, and really delighted at the work that you're doing with the rest of our colleagues across UNC School of Medicine and others on READDI. Take a lot of comfort, as Dr. Burks said, that you all are really thinking about this next wave of what's to come. So as we close out our time together, I just want to say thank you so much for taking the time to be with us. We've listed a few resources on the screen here if you're interested in getting more information. And most importantly, if you've been moved by what you've heard today and you'd like to support the READDI project, there are ways to do that listed on the screen, as well as our COVID-19 response fund. So Thank you again for your presentation, Nat, and for Dr. Burks, your wonderful hosting abilities. And then I just want to say, I hope you all will mark your calendars for our next Game Changers session, which will take place on June 30th. This time we're going to be talking about cancer and our CAR-T program, our cellular immunotherapy program here at UNC. So we'll be joined by Dr. John Cerroti to do that. So I will leave you all today then with just, again, many, many thanks, and we'll look forward to seeing you again next time.
Thank you all for having me today.
Yeah, Nat, thank you. That was great.
Thank you, Nat. Thank you, everyone, and have a good afternoon.